Okay, let's proceed to the next talk. It's similar regarding parasites this time. So we will discuss the role of parasites in remodeling of microbiota and in colonic neoplasia. Dr. Messaritakis is a senior scientist at the Translational Research Laboratory at the Medical University of Crete. Dr. Messaritakis.
Good morning. Thank you very much for the kind invitation. Well, before proceeding to the role of parasites in remodeling microbiota, I would like to very quickly to mention again some important facts about gut microbiota itself. Human gut represents a complex ecosystem composed by large microbial community regulating some important functions. The species composition varies among individuals and may change over time. We all know that what happens in Vegas stays in Vegas. However, this is not the case for the gut. And as Hippocrates said, all disease begins in the gut. Indeed, dysbiosis has been associated with multiple diseases and is considered of a major public health importance. The mechanisms are not known. However, healthy individuals have greater microbial diversity. Therefore, it is important to investigate the factors that influence the natural healthy gut microbiota before analyzing the consequences of dysbiosis. There are many factors that affect gut microbiota. Clockwise, from top left, is the methods of delivery at childbirth, whether breast or bottle feed, diet, exercise, general lifestyle, diseases, especially intestinal inflammation, aging, drugs, especially antibiotics and antacids, and finally geography and cohabitation with animals. An important limitation of the current studies is actually the disproportionate focus on industrialized populations. Indeed, improvements in sanitation and medication have led to decreased exposure and transmission of microbes. And as a consequence, these populations have a reduced parasitic burden than our ancestors. Human parasites are generally classified into two main groups, protozoa and helminths, which are further divided into tapeworms, roundworms, and blood flues. Notably, industrialization has been correlated with the nearly complete disappearance of these parasites. Now, the question is whether these gut eukaryotes are simply parasites that are detrimental to the human health, or they could also provide some benefits. Moreover, we need to understand what is the role of these parasites in shaping gut microbiota. To answer this, we need to focus more on the microbiota in non-industrialized populations which still have high prevalence of these parasites. Historically, protozoa and helminths have been considered as parasites that are detrimental to the host organisms. However, there are still commensal and or beneficial parasites. And indeed, clinical improvements have been shown that individuals with diarrhea or bowel inflammation have led to the idea of helminthic therapy. In a recent study, the presence of commensal protozoa other than the pathogenic one, Entamoeba histolytica, have been associated with an increased microbiome diversity in a non-industrialized population. Now, to answer whether these human parasites are friends or foes, we need to find out whether the increase in microbial diversity is associated with protozoa in general or only with the non-pathogenic ones. We also need to understand whether the effect of protozoa on microbial diversity is mediated through bacterial parasites interactions or indirectly through the host immune system. In a recent review article, the authors revealed the presence of parasitic or commensal parasites of the human intestinal tract. Now, concerning the effect of pathogenic parasites, In a study, it was shown that utilizing a number of various models revealed no clear link between the presence of Giardia intestinalis and microbial diversity. In another study, Entamoeba histolytica was correlated with a decrease in various microbial species and with an increase in Bifidobacterium species only. Finally, in another study, in a group of asymptomatic children infected with Entamoeba histolytica had a lower abundance of Prevotella cori than the symptomatic ones, providing a complex interactions between parasites, microbiota, and the host. Now, concerning the effect of commensal parasites, the studies showed a negative correlation between the presence of Blastocysts and Bacteroides species and a positive correlation between Blastocysts and Prevotella and Ruminococcus species. In another study, Blastocyst colonized patients found to have a higher abundance of various microbial species, whereas Blastocyst-free patients were raised only in Enterobacteriaceae. And finally, the presence of intestinal Entamoeba species, other than the pathogenic one, associated with higher microbial diversity. Now, human parasites are very well known that have carcinogenic properties, with inflammation being the most proposed mechanism. More specifically, for the case of colorectal cancer, its relationship of Schistosoma japonicum infection was initially mentioned in the English literature in the middle of 1950s. Since then, a large number of patients with colorectal cancer and schistosomiasis were reported individually or in groups and are mentioned here. The authors attempted to correlate the mortality due to colorectal cancer and due to schistosomiasis, and most of them found a positive and significant correlation. In one of these studies, the authors compared colorectal cancer mortality and schistosomiasis mortality due to Schistosoma japonicum in two areas in eastern China, and found a high prevalence of schistosomiasis that ranged between 10 to 79%. There was a significant correlation between these two mortalities. However, there was no statistical relationship between mortality due to lung cancer and due to Schistosoma japonicum. In another study, the authors found schistosomal eggs more likely in patients with stomach or colorectal cancer. Then, a large number of resections of the GI tract were reviewed and found a significant correlation between malignancy and schistosomiasis. Interestingly, patients with malignancies and Schistosoma japonicum infection tended to be younger, and this was also mentioned elsewhere. In another study, the authors attempted to compare a number of schistosomal and non-schistosomal associated colorectal cancers, and found that the schistosomal colorectal cancers tended to be more often well differentiated. Now, evidence of colorectal cancer and Schistosoma mannsoni infections have been reported for decades, primarily in the Middle East. However, in one of these studies that included a large number of patients from an endemic area of Schistosoma mannsoni underwent lower endoscopy and biopsy. However, only 2 patients had evidence of both Schistosoma mannsoni and colorectal cancer, thus leading the authors to conclude that there was no link between Schistosoma mannsoni and colorectal cancer. In another study, the authors compared again schistosomal and non-schistosomal associated colorectal cancers, and found that the schistosomal associated colorectal cancer patients tended to be younger and often had multiple synchronous tumors. Moreover, pathologically, the tumors were often mucinous and tended to present at higher stages, while overexpressing p53. Finally, in another study, schistosoma manzoni eggs were more commonly found in MSI high colorectal cancer patients and in KRAS mutant patients. In conclusion, carcinogenesis associated with parasitic infections is a complex process that involves several mechanisms, but definitely chronic inflammation. Among all species, Schistosoma japonicum has been more linked to colorectal cancer, but despite the weak evidence, some reports also associate Schistosoma manzoni with colorectal cancer. There is no strong evidence that links GI neoplasia with any other parasites besides schistosomal species. And now the question is whether if there is simply no relationship between most GI parasites and neoplastic complications. Perhaps, but maybe this is just a statistical issue. And to answer this, larger epidemiological studies are required to see if there is indeed an increased risk. Thank you very much.
Thank you, Dr. Messaritakis.
No, it's for the previous.
For the previous. Okay, are there any questions from the floor?
Phil, you have to answer the question. That's for you there.
So yes, they can. Well, they actually see the bacteria in the metastases, and so the question is, what's going on in those metastases? What's the relationship between the fusobacterium and the cells? Is it symbiotic? Is it causative? We just don't know. So it's there, and we need to understand it much more because. Can we influence the metastases by giving antibiotics? There's evidence in cell culture that when you co-incubate fuso and cancer cells and you give antibiotics, that you can reduce the rate of the growth of the cancer cells, but there's nothing in humans as yet.
Dr. Messaritakis, what is the prevalence of parasitic infections in the Western world? I mean, is it you present studies?
From Middle East actually, yeah. There are not many studies that have been performed in the Western world actually. So this is a good chance actually for us to start something about that.
Okay. There's no other question. Okay, let's proceed with the